Hi guys, welcome to my YouTube channel Raise Science Simple, and let's make the science simple. And I'm giving lectures currently on the CRISPR. So today we are going to discuss about the overall CRISPR protocol. So first of all, what you have to do, you need to design the sgRNA that is against your template strand. And your sgRNA should not contain the PAM sequence. So the one thing which you need to remember is it should not contain the PAM sequence. So number one thing which is important is should not contain PAM sequence. Okay. Number two, it should be complementary to your template strand. Okay. So these are the two things which you need to uh, keep in your mind whenever you are designing your experiment. Now you design the complementary sequence here because you need to clone this sgRNA in the plasmid. And if suppose you are using the plasmid which has the U6 promoter, then in that case you need to add CACC at the 5' portion and the AAAC in the reverse strand at the 5' position. And again you have to add the G, because if you add the G nucleotide at the 5' position, the efficiency of the transcription basically increase if you are using the plasmid that has the U6 promoter. So here you can see this is the plasmid that has the U6 promoter, and we have digested it with the BSMV1 enzymes, and therefore it is producing these overhangs, and that is the region we are adding CACC and AAAC here. So you can see here CACC and AAAC here. Okay, after that what you are going to do, after you are done after you are done with your designing of sgRNA, and then you need to clone your sgRNA in the number one step is clone your sgRNA in the plasmid. You can design your sgRNA in such a way that it has some reporter molecule if you want to just see under the fluorescent microscope. Now you need to clone sgRNA in a plasmid. Once you have cloned it, after you have cloned it, you need to isolate the plasmid DNA and select only those clones which has your sgRNA. So this is your number one step. This is your number two step. And number three step is select the clones that has the desired, that has the sgRNA. Okay. It means that is not empty plasmid DNA. Now fourth point what you have to do, now you need to do the transfection of your cells with this plasmid DNA. Transfection of cells with plasmid DNA. Okay. So once you will do the transfection of cells with the plasmid DNA, what it is going to be there? If suppose this is your plasmid DNA, and again it is double stranded, remember that, okay? And this is your, let's say this is your sequence, which is your sgRNA sequence here. So this is your sgRNA sequence, and this is your plasmid DNA. So now you are doing transfection in the cells. Now what will happen in your cells? There will be the RNA polymerase. Okay. So this RNA polymerase and this sgRNA should be upstream to the promoter region. So let's say this is your promoter region. This particular is your promoter region. just upstream of the sgRNA sequence. So this RNA polymerase, basically what it will do, please listen very carefully because it's a critical point. So once this RNA polymerase will bind to this promoter sequence, it will initiate the transcriptional process by using this strand as the template strand. So what it is going to do, this strand is going to work as the template strand, and therefore this RNA polymerase will produce the complementary sequence of this template strand, which is again same, will be having the same sequence as the sgRNA. So what you need to keep in mind in this particular thing is this plasmid DNA is double stranded. You are doing the transfection, then the RNA polymerase of the host cell will recognize one strand as a template strand. And how it is going to be recognized? So your sgRNA should be near the downstream of the promoter region. So RNA polymerase will bind to this promoter region, and this particular sequence, another sequence, it will utilize as a template strand. Okay, so this RNA polymerase will bind to this template strand, and therefore it will produce the complementary sequence of this template strand, which will be nothing but the sgRNA. Now what will happen? Let's say after the transfection, 5 number point is this: you need to select the clones, select the cells which has your desired edit. Okay, and that you can do by either you isolate the DNA and you do the sequencing, or next generation sequencing, or the Sanger sequencing by using the primers, which will flank your sgRNA sequence, and that is automatically being designed by the ChopChop software. So you can use those primers for sequencing. Or what you can do, if you want to see the functional aspect, you can isolate the protein and do the Western blot, or you can do even the flow cytometry to see the expression of a particular protein if you want to see the functional aspect of your edit or the mutation. So this is like the how you can do the selection by the sequencing or by the Western blot. NGS also you can do, next generation sequencing, or the Western blot, you can do that. And another method which you can use is this one, T7 endonuclease assay you can do. So in this, what you are going to do, you amplify your products, denature and renature, and the T7 endonuclease only cut the heteroduplex DNA. I have already explained in my previous lectures, so please watch those lectures. So it will cleave only the heteroduplex DNA. So in this DNA, this and this, it is going to be cleaved, but not these 4 DNA. Either they have the mutation in both the strands, or either they are not mutated at all. So how will you differentiate it? So for that you should add the control sgRNA. So this sgRNA will interact with this, because you will denature and then re-anneal, and in that case it will be forming the heteroduplex DNA, which is going to be cleaved by the T7 endonuclease. So then you can say that this particular region, in this particular cell or in this particular clone, both the strands are mutated. So these are the things which you can do for the selection. And the one thing what you can do here, when you are done with the transfection and you isolate the cells which has the desired edit, you can do the serial dilution. In the serial dilution, that is your optional step basically. So in the serial dilution, you take the cells which has your desired edit and you dilute it so that you get the single cell. If suppose this is your 96 well plate, you do the serial dilution, and in the last well you might be having only the single cell, and now you isolate the DNA from this cell and do the sequencing, or you can even put this cell in the 96 well plate again so that it grow basically. So that's how you can do the CRISPR protocol in the laboratory. These are the some basic steps. So that's all about this lecture, and if you like the content of my lecture, kindly subscribe my channel. Thank you so much.